Hello, welcome to today's biocast. Today we're going to talk about meiosis I. Now we've had biocasts on mitosis, and remember mitosis was responsible for producing any type of a cell in your body other than a sperm or an egg. So every other cell that you make is the product of mitosis. Meiosis, on the other hand, is responsible for creating the sperm and the eggs. If you remember, you as a human are a diploid organism. That means you have two copies of each chromosome, one you got from mom and one you got from dad. In order to make a baby, that we have to reduce the number of chromosomes in the sperm and egg from two copies of each chromosome down to one. So meiosis is often called reduction division. So let's take a look at how this process is going to work. One of the things you'll notice is that meiosis and mitosis, the various stages of the replication and division of the DNA, have the same names. They're prophase, metaphase, anaphase, and telophase. You can tell the difference of whether you are in meiosis or mitosis by whether or not there is a number after the name. So prophase 1 means that we are going to be in meiosis. Anytime there's a number after the word, after the stage, we must be in meiosis. There's two stages to meiosis, and we'll have another biocast on meiosis II. So let's take a look at what's happening in prophase 1. In our cell right now, we have our centrioles at the poles, the centrioles which are going to pull the chromosomes around throughout the various stages of meiosis. First thing we're going to do in prophase is we're going to condense out our chromosomes. So we're going to condense out one chromosome from mom and another chromosome from dad. But remember now, part of meiosis, just like in mitosis, is that we have to duplicate our chromosomes. So we're going to make a copy of each one of these, a copy of our mom's chromosome and our dad's chromosome. We still only have two chromosomes. This entire structure is called a chromosome, and this entire structure is called a chromosome. Each one of these chromosomes is made up of two sister chromatids, though. This is a sister chromatid, and this is a sister chromatid. So if we take a look at it, I'm going to animate our sister chromatids for a minute. One sister chromatid, a second sister chromatid of that chromosome, which we said came from your mom. Down here, we have a sister chromatid which came from dad, another sister chromatid which came from dad, and together they make up one chromosome. Obviously, though, we're going to have to do this for all of our chromosomes. Now, we're not going to fill out the whole complement of human chromosomes. There's 23 of them. That would fill this slide up pretty well. So we're only going to look at 3. So we're going to take a look at our blue chromosomes. We're going to have red chromosomes, and we're going to have our green chromosome. What we have is 3 different chromosomes, but we have 2 copies of each chromosome. So we have 6 chromosomes in this slide, and we have 12 sister chromatids in this slide. We have 1, 2, 3, 4, 5, 6 chromosomes. Remember though, this chromosome, the blue chromosome here, and the blue chromosome here are homologous. They're copies of the same chromosome. They're both the blue chromosome. But now remember, we said we had 12 sister chromatids in here because each chromosome is comprised of 2 sister chromatids at this point. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and our last 12th sister chromatid. What we need to do now, though, is we need to move these chromosomes onto the metaphase plate. And that happens during metaphase. Metaphase 1 to indicate we're in the first round of meiosis. So let's move everybody to the metaphase plate. These guys move, and you'll notice they have lined up in a different fashion than they did in mitosis. In mitosis, they lined up with each chromosome running along the metaphase plate here. In this case, what we've had are two chromosomes line up on either side of the metaphase plate. One copy of our chromosome above the metaphase plate, a second copy of the chromosome below the metaphase plate. One copy of the chromosome above the metaphase plate, second one below. One copy of the chromosome above the metaphase plate, the second one below it. This is in contrast to what we saw in mitosis. This structure of having our two chromosomes lined up on top of each other, this structure is called a tetrad. We have three tetrads here. We have tetrad 1, tetrad 2, and tetrad 3. Now, let's get our numbers in order again. Just be like the count on Sesame Street. We have 3 tetrads. 1, 2, 3. We have 6 chromosomes. 1, 2, 3, 4, 5, 6. And we have 12 sister chromatids. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now what we're going to do in the next phase is we are going to break these tetrads apart. Pull one of these chromosomes towards the North Pole, the other one towards the South Pole. So we're going to break our tetrad apart. Here we are in anaphase now. We have our microtubules that are coming out of our centrioles and attaching to our centromeres, remember, just like we had in mitosis. And those microtubules are going to pull the chromosomes towards their respective pole, just like they pulled everybody into alignment along our metaphase plate. So let's see what happens. So we've ripped apart our tetrads. So now we have 0 tetrads. We still have 6 chromosomes. We still have 12 sister chromatids. So anaphase 1 is complete. We pulled our chromosomes to the pole. Next thing we need to deal with is telophase 1. Telophase 1 is where we start to separate the one parental cell to create the 2 daughter cells. If you remember, we can tell the difference between plant cells and animal cells based on how they divide in cellular reproduction. Plant cells start to form a wall internal to the cell. So we start getting segments of our cell wall forming, and they keep forming until eventually they make a complete cell wall and have isolated our 2 cells. In contrast, when we look at an animal cell, we have a belt of microtubules that run around the cell, and just like if you had a balloon with a string wrapped around it, as you tighten that string, it separates the balloon into 2 pieces. Same thing happens here. As we start constricting the microtubule that's around the cell, it's going to pinch in this cell, eventually creating 2 unique cells. So we're going to see this pinching in of our 2 cells, and it's going to keep getting more and more pinched until eventually it seals it off and we end up with our 2 cells. I hope that helps you understand Meiosis I. We'll have another biocast on Meiosis II. Remember though, meiosis and mitosis are very, very, very different processes. They look somewhat similar. They have the same names for stages, but they are incredibly different. Mitosis produces all of the cells in your body except for sperm and egg. Meiosis produces only sperm and egg.